Okay, so now I'm going to cover on RFLP, restriction, which stands for restriction fragment length polymorphism. So this is one thing that it was actually covered in term 1 biology, but it seems that when it came to term 3, many people can't remember, and this was not recapped during the core sessions or the lecture. So it seems that a lot of people have been struggling to know what this RFLP stands for. Okay, so restriction fragment length polymorphism actually means that actually it is a state, more like a symptom that we observe when two people have different alleles. Okay, so for example, let's say this individual A and this individual B. So this guy has brown eye, this is phenotype, and this guy has blue eye. Okay. So the thing is that so this guy is going to have allele for brown eye and allele for blue eye for this guy. And so what makes the difference in allele is that they're going to have a difference in usually a single nucleotide, and this is what people call SNP, or SNP, which people call, which stands for single nucleotide polymorphism. So in other words, the difference in allele comes from difference in one single nucleotide. Yeah, so for example, if we zoom this part in, let's say the individual A has the nucleotide sequence AATCCGA, okay? And at the exact same loci, which means the exact same location of the DNA, this individual B has AATTCGA. So there's a difference of TC and TA. Okay, so this is a SNP as a single nucleotide polymorphism, which leads to the RFLP. So now, what exactly is RFLP? Then I first have to explain about restriction enzyme. So restriction enzymes are enzymes that cleaves, which means cut, a sequence of DNA at a specific spot. So for example, so these points are going to places that the restriction enzymes actually cut. Okay, so so let's take a look at this. So in this case of AATCCGA, the restriction enzyme, I'll call this restriction enzyme ABC. Okay, so it's just a random name I came out of. So this restriction enzyme ABC will look through, and if it finds the sequence AATCCGA, then it will cut at the spot here, for example. Okay, so this cut will be the point of cutting at this point. And let's assume that there's another restriction enzyme, DEF, and these will be cutting at these spots. Okay, and same for this one. Okay, so what makes the difference in okay, so what this RFLP means is that the difference in allele means that there's a difference in single nucleotide makes the restriction enzyme ABC unable to find this sequence. Okay, so when it is AATCCGA, the restriction enzyme ABC can find it and actually bind to it and actually cut at the spot. But when it is AATT, CGA, then it can't find. It's like, oh, this is not the one I'm looking for. So it's just going to bypass and look for the sequence it wants. So in other words, after we have added this restriction enzyme ABC, this person is going to have cut once, twice, and thrice. But in the case of individual B, this person will only have cut twice. And by the way, so these things, the DNA that has been clipped out, is called restriction fragments. Okay, so this what gives us to the restriction fragments, and the difference in allele leading us to able to use the make use of the state RFLP. Okay, so let's assume that from individual A, the restriction fragment we're going to get is with the length of 20,000. Okay, it's 20,000, but we'll just simplify it. 20,000 and 50,000, and from individual B, it is going to be 70,000 base pairs. Okay, and okay, I hope you recall what gel electrophoresis was. Yes, and just in case you don't recall what gel electrophoresis is, I'll put the link to the notes in the description. Okay, so in this case, we're going to have the wells here, and so for this one is for individual A, and this one is for individual B, and this one will be the ladder. Oh, just in case you don't know what ladder is consists of like machine synthesized DNA fragments with the specific length. So for example, okay, of course, when we at the start, it's all going to be we'll just have one well, like the same as others. But as we start the recording, then it's going to progressively go on and onwards. And since the shortest one can pass through the agar cell the fastest, it will be at the furthest. It will be furthest away. Okay, for this individual A, this guy is going to have a restriction fragment with the length of 20,000. So in other words, he's going to have one bar here and another one at 0.50. So he's going to have another one here. For individual B, he's going to have a single one, which with the length of 70. So he's going to have one bar here. So through this way, we can see that the restriction fragments of this person A is going to be much shorter than B. In other words, we can actually see that this restriction fragment A, not restriction fragment, restriction enzyme ABC from just now is will show that whether what kind of allele this person has. Okay, so I'll add in some additional explanation with sum up. Okay, so difference in allele arises from usually from difference in single nucleotide, which is called single nucleotide polymorphism, SNP, SNP, and this allows us to use a certain restriction enzyme to differentiate these. Just for information, usually we have the information that this restriction enzyme will bind at specific spot. Okay, so for example, just now restriction enzyme ABC will be sure to bind to the nucleotide sequence AATCCGA, which we know that that sequence is on the allele coding for the eye color. Okay, so the difference in the nucleotide sequence makes restriction enzyme possible to bind on certain allele, but not on other types. So this will lead to a different length of restriction fragments. And if we do gel electrophoresis out of these, we can see that different restriction fragment length through this the gel electrophoresis, and through this we can see that whether this individual has this allele or not. Okay, since the method of Southern blot was mentioned inside, I'll explain what exactly Southern blot is. So Southern blot is the method to see the difference in refraction length of refraction fragments. Yes. Of course, yes. If you have tried it in the lab, you can see that we actually put in dye within the DNA, but we usually use this method of Southern blot to find out like specific restriction fragment that we wish to look for. Okay, so if I explain the entire process, please recall PCR, polymerase chain reaction. So which one is to amplify the number of the restriction fragments we have. And if you can't remember what PCR was, please refer to the notes behind, not behind, in the links stated below. Okay. Okay. So after PCR, we're going to get these strands length of DNA, and we're going to put this in gel electrophoresis. Okay, I hope you recall what your electrophoresis because it's in with like at the front of this video. So if you can't, it's seriously a problem. Okay, so this we're going to get the different refraction lengths in the agarose gel, and this time around we're not going to add in any dyes or any of those kind of things. Okay, so it's technically we really can't see them in with naked eye. So what we do next is that uh, we're going to have we have to imagine this. So we're going to have a plate, and we're going to have salt water inside. Well, salt solution to be exact, but never mind. Okay, and then we're going to have a layer of sponge. Okay, then we're going to have our agarose gel. Okay, so this thing is going to come here. On top of this, we're going to have this nitrocellulose paper. It's not okay. It's not really important, but just in case you might be curious. Nitrocellulose paper, and on top of this, we are going to put paper towels. Why paper towels? Okay, so this is what happens. Okay, when we put this inside the salt solution, so salt solution is going to enter the sponge. It's going to move up through the gel, and through the nitrocellulose paper, and to the paper towel. In the process of moving, the restriction fragments that is inside the agarose gel is going to follow the salt solution until the nitrocellulose paper. So it's going to be the restriction fragments are big enough to pass the agarose gel, but small enough to pass through the agarose gel, but too big to pass through the nitrocellulose paper. But salt solution will continue moving outwards because it's you can think of it like water, which is yeah. So what we do next is that we get this nitrocellulose paper. So what we do to this is that on this we're going to get the exact same how should I say, the patterns we're going to see as what we saw in the agarose gel. Okay, so what happens is that they're actually like stuck. The restriction fragments are stuck on the nitrocellulose paper. So and these are going to be unpaired state. Okay, so it's going to be a single-strand DNA. And what we're going to do is that we're going to put this inside a plastic bag, and we are going to insert also this thing called probes. So these probes are also single-stranded DNA, which is going to be exactly complementary to the DNA strand that we're looking for. So for example, if the strand we're looking for is AATCC, then we're going to have TTAGG. So this is going to be our probe. Of course, it's actually going to be much longer in real life, because if it's too short, it will like just bound everywhere. Okay. So anyway, so what is going to happen is that these probes are actually radioactive. Okay, it's like literally radioactive, and these probes will be so inside this plastic bag. We're going to put in a solution with the probes, and then these probes will hybridize. Remember the hydrogen bonds forming between the nucleotides? Yes, and hybridize with the restriction fragment that we want. For example, if the restriction fragment we want is let's say this one, okay, the one I'm highlighting blue, then it was only going to hybridize there. So what we do after this is that we take out the nitrocellulose paper, we wash it with a certain solution, which I can't recall at the moment. Yes, and then the reason why we wash is that we remove so that we can remove the like excess um, radioactive probes that is on the surface of this nitrocellulose paper. So that is the one that has been bound to the sequence we want. It's just going to stay there. Okay. Alright, so after we wash the thing, we're going to expose X-ray on it. Yes, the reason why we expose X-ray is because those that is radioactive is going to like how should I say, react to it, and not react to it. It's like it's we can see the light from it, so we can actually if we do a X-ray, we can actually see the exactly the probe that we wanted. So if we match like the location of it, then we will be able to find out. Oh, so this is the one that has the exact DNA sequence that we want. So the method of Southern blot is to find the DNA sequence. In other words, which can be allele, which is to Southern blot is to find a specific DNA sequence or allele that we want to find. Yes. Oh, and by the way, if the probe is, if it's not just one, but if multiple types that different kinds of we want to see, so it can actually also bind to others as well, which will be seen in others as well. Yeah. So that is the process of Southern blot, and that will be the end of this video. Thank you guys.